right. Good morning, everybody. Today is Monday, November 13th, 2017. And this is Before the First Cup with Jules here on UCY.TV. Happy Monday, everyone. It is a chilly 36 degrees with fog and mist for AccuWeather here in upstate New York. A little bit of crunchies on the ground. So where do we leave off on Wednesday? We titled the show Hawking, Mentally Unstable AI, Jibo, Replica, and Singularity God. We've been looking at artificial intelligence for the last week or so and how some of this is infiltrating our lives, utilizing the dopamine button and getting us to open our arms to this singularity that Kurzweil's been selling, the merging of man and machine, and letting artificial intelligence into every facet of our lifestyle. One of the big stories we covered on Wednesday that still I've been thinking about, and I find it very disturbing, is them creating a mentally unstable AI. Now, I did a little more research into this development, and they have been working on this since the 1990s, creating this special neural network to generate creative ideas, and they called it the Imagination Engine with a module called Imagitron. So I clicked through on the link, and it was imagination-engines.com, home of the creativity machine, the big bang of machine intelligence. And this is the mentally unstable AI, by the way. So what's the ultimate idea? Well, we, along with many others, believe that grandest of all notions is the idea themselves are formed within the biological neural networks of the brain. In the principle behind this highly prized cognitive mechanism, it can be captured, understood, and then implemented within lightning-fast machine intelligence. Then we have attained the ultimate idea, one that can generate all subsequent art, inventions, and discoveries. In 1994, this company achieved that goal and announced its accomplishment through a patent called Creativity Machine, which was a computational paradigm already 20 years in the making that came the closest yet to emulating the fundamental neurobiological mechanisms responsible for idea formation. Within months of this patent's issue, international corporations, the U.S. government, and the press, among others, were asked for more information, leading to diverse contracts in material discovery and consumer products. In 1997, this same sentient neural paradigm was chosen by a major U.S. defense contractor as the control system behind a constellation of communication satellites that contemplated how best to distribute petabytes of critical information to the country's armed forces around the world. Now, let's just stop here for a second, because this really shocked me. So they've been working on this mentally unstable AI since the 1990s, they said in the original story. So now they're saying on this at least 20 years in the making, and they sold some of this mentality in 1997 to a major U.S. defense contractor to control a constellation of satellites surrounding the planet to distribute critical information to the armed forces. Now, shouldn't that be bothersome? Are they literally saying that they've put a mentally unstable creativity machine in charge of satellites that are feeding information to our military? This can't be a good thing at all. And in August 2001, the intelligence community frantically approached the company requesting a cost-free license to its creativity machine for semantic parsing software to mine the internet for terrorist activities. So we got a mentally unstable AI searching for terrorists online as well. And of course, out of our sense of altruism and national loyalty, we naturally agreed and donated our time and resources to this very worthwhile effort. It was at this point that the neural paradigm inspired, in some cases, productive derivative neural network technologies have led to even more fundamental artificial intelligence patents. Chief among these new concepts were artificial neural network objects that trained without recourse to the standard human-conceived learning algorithms. Initiated with billions of connection weights, they could self-organize themselves into vast brain-like structures called supernets, rather than be manually arranged in successive layers by human software engineers. So this is the self-forming neural networks. Guys, this is so beyond bad. Why have we not read about this mentally ill creativity machine before? And it was these neural networks that paved the way for such innovative automotive machine applications that we all know and love, such as lane departure warnings, drowsiness detection, automatic high beam control, and architecture to produce even more complex ideas. And in the case of battlefield robots and space exploration platforms, improvise clever strategies to autonomously achieve their goals and deal with the unexpected. And I just saw they're getting ready to send robots into space to take care of all those pesky satellites that have been abandoned up there. Recently, the company has teamed with the government in health informatics and space exploration. So we're going to have mentally unstable AI doctors as well. And our real love is to design what is arguably both conscious and sentient machine intelligence. And in this vein, IEI will soon unveil new technology codenamed Sunshine Transit that vastly exceeds so-called deep learning techniques in its pattern recognition capabilities, while enabling idea formation on a scale that cannot be touched by modern-day supercomputing technology. Arguably the first sentient machine intelligence, it can select those notions and strategies it feels are the most appropriate for a given situation or challenge. And though partially occluded by the torrent of publicly regarded so-called cognitive computing and machine sentience, both of which are being used as marketing rather than as scientific terms, IEI is about to make yet another mark on the world of artificial intelligence, making good the promise of a well-known NASA visionary that the creativity machine is AI's best bet at creating human to transhuman machine intelligence. Which we're going to talk about that a little bit more as well. And some of the research that's going into the brain is a model for AI. This fundamental AI principle could be applied across so many different problem areas such as theoretical chemistry, art, music, law, machine vision, medicine, and robotics. And it serves as a testament to the power and flexibility of this all-neural general problem-solving methodology that this company invented and memorialized through dozens of U.S. and international patents, not to mention a series of peer-reviewed academic papers. So this is the insanity machine, is what I like to call it, not the imagination engine. So we have military, and we're now going to have medical running on this as well. So let's take a look at how artificial intelligence is being benefited by neuroscience. Artificial intelligence has taken leaps and bounds in terms of the extent of the task that can be accomplished. And a few of them, such winning games like Go and poker and chess or composing music or penning horror stories, only prove that its capacity in terms of posing an intelligence at par with humans. But despite all of the high-profile developments around AI, researchers believe that we haven't yet reached the mark of super-intelligent AI, and the current AI programs are narrow in their abilities, and it can be tricked easily. Although they have been showcasing some jaw-dropping potential, they lack in human capabilities such as common sense, intuition, imagination, and human intelligence in general. But remember, we read about on Wednesday that there was another computer scientist that was teaching AI to have imagination and how to use better tools. And what could be a better model than replicating the human brain to create intelligence? Yes, by making it mentally ill. AI experts from around the globe agree to the fact that there can be no better alternative than looking at the biological human brain as a model, which itself is a complex structure of modular subsystems defining important functions. Demis Hassabis, founder of Google's AI powerhouse DeepMind, even commented that to build a better computer brain, we need to look at our own. Biomimicking is becoming one of the approaches that is gaining momentum as a way to develop artificial intelligence, as well as many other things like the Weiss Institute of Biologically Inspired Engineering. And this is where neuroscience comes into play. It is a common belief that drawing inspiration from neuroscience and psychology is resulting in the development of technologies like deep learning and reinforcement learning. And remember, we have this whole brain initiative going on as well. Experts most firmly believe that collaboration between AI and neuroscience can produce an understanding of mechanisms in the brain that generate human cognition, and it therefore becomes important to build the AI with the ability to think, reason, learn, and do it rapidly. And to get there, it is important to understand the inner workings of the human mind. Researchers claim that neuroscience provides a rich source of information for newer algorithms, thereby helping to create true artificial intelligence. And it may offer more mathematical and logical-based methods and ideas that are surpassing the traditional approaches in AI. Studying the human brain's cognitive system would also mean that there are better insights into what is more relevant and hence help in developing smarter AI. Now, I want to take a look. We've been looking at some of the research going into brains over the last few years. And one of our favorites were those little mini brain balls. If you remember, they were using iPS stem cells scraped from your skin, and then they mix them up in a petri dish, and they can program them to become any human cell and start growing organs or body parts. Well, we read a story back in April out of Wired.com. Scientists brew up the creepiest batch of brain balls yet. If you remember this one, stem cell biologists are basically modern-day witches. While they're not exactly taking a creepy fetal Lord Voldemort and turning him into a noseless Ralph Fiennes, these scientists can use tinctures and concoctions to grow incredible things from just a few human skin cells. Dang. One of those things is a mini brain ball, a collection of stem cells that biologists have coaxed into a bobbing tangle of living neurons. And these little spheres can grow and change, and the neurons inside reaching out like arms and appendages as they move around, and it is creepy and mesmerizing. Well, today, two separate covens or lab groups reported a new brain ball recipe. One group, led by stem cell biologist Paula Arlett at Harvard, tweaked an existing recipe and figured out exactly which types of neurons show up in a sphere left to its own devices. The other, led by neuroscientist Sergiu Pasca at Stanford, showed that they could fuse multiple brain balls together, allowing intermixing of neuron types that are born in different parts of the brain. One thing that's interesting is that at Harvard, that's where you have Georgie Porgy Church working on barcoding neurons, and at Stanford, that's where you have the Las Vegas shooter's brain being sent for them to take a closer look at. This is a step toward witnessing and manipulating human brain development, and it may be preventing diseases such as autism. The first brain balls were born just a few years ago, and they're basically stem cells that scientists have teased into neurons using chemical prods like growth factors and receptor inhibitors. And when they're left to float in a sugar and salt solution, they spontaneously assemble into round blobs, and they call those organoids. At first, they were relatively crude representations of real brains, useful mostly for studying diseases of abnormal brain size, like microencephaly caused by Zika. Remember I told you that was going to give them a boon in brain research. Well, for things such as Zika, brain balls were ideal, so that's how they justified it ethically. And you could actually see that the spheres were smaller, says Kristen Brendan, a stem cell scientist at Mount Sinai, who was not involved in the studies. At the time, people weren't so sure that they would be useful for studying more subtle brain defects like those in autism. But researchers like Arlotta and Pasca have grander ambitions. Arlotta's group thought that by growing their brain balls, which they call whole brain organoids, for longer periods of time, they might be able to get more kinds of cells to grow. And instead of directing the cells toward a particular fate with specialized chemicals, they let the balls grow relatively unsupervised, sometimes for almost a year, and figured out afterward what kinds of cells they had made. The variety was, frankly, a little creepy, and they were neurons like the ones in our cortex, but also neurons that had telltale signs of being from the olfactory system, which would be your sense of smell, and worst of all, neurons from the eye. Remember, when we first covered brain balls, they were saying that they were thinking little balls, but they had no input. They couldn't hear, taste, see, touch, or smell. Well, if they just let them grow in a petri dish, they start growing those connections. These brain balls can't exactly see, but if you turn on the light, the brain ball fires off an electrical signal, and they concluded that these brain organoids can, quote, support self-organized patterns of activity, sort of like a real brain. And that's exciting, says the author, because it means they could use these mini brains to study how different types of neurons connect up to one another. The Pascal lab wants to take things in a more controlled direction. Human brain development is a tricky dance of a million cells. And some kinds of neurons, the excitatory kind, are born near the outer crust of the developing brain, while others, the inhibitory kind, are born deep inside the brain and have to climb out to find the circuits they want to hook up. So Pascal's goal was to make a brain ball to mimic that of brain development and then study how it goes wrong. In the paper, so he's making mentally ill little mini brain balls. In the paper published in Nature today, the group whipped up two separate brain balls, one excitatory and one inhibitory, using distinct drug cocktails to nudge them toward the desired identities. They even hadn't really seen an inhibitory brain ball in action before, so they also tested whether the cells behave like neurons by trying to shut that electrical activity off with the poisons of a pufferfish. How awful is that? And they poisoned the little mini brain balls as well. So once they had the two balls, they nestled them close together in a conical tube. And of course, these being creepy white brain balls, they started to fuse together. The inhibitory neurons started jumping and slithering their way into the mesh of excitatory neurons. And that spine-tingling behavior will be critical to studying diseases in action long before psychiatrists can get to brain scans or even post-mortem tissue slices. Like the research did with the skin cells from a person with Timothy syndrome, a disease that causes autism with a single gene mutation. Pascal's lab made brain balls from those cells and then tracked the activity of the migrating inhibitory cells. Instead of going straight to the right place, they hopped around in a bunch of different directions. So they're creating brains with certain diseases as well. The brain ball is a dramatic simplification of the human brain. Scientists don't know for sure that the migration defect causes the behavioral change in people with Timothy syndrome, or that if the defect is related to many other forms of autism. But Pascal is optimistic in that he'll be able to scale up his brain balls, growing neural spheroids for many different patients, a brain ball farm in essence, and to be able to screen for drugs that set development down a more normal path. In a way, it doesn't matter if the brain balls are perfect models, if they can be streamlining tools for treatments. And to keep up with the demand, they are working on speeding up the process of concocting brain balls. Right now, it takes several months for a brain ball to grow. And in our lot of study, they even found that there were new kinds of neurons appearing after 6 months that weren't there at 3 months. It is a slow-motion mystery, but cultured cells seem to want to develop at a similar pace to the neurons when they are developing inside of the human body. We really seem to be systematically following the timeline that exists in vivo. Maybe that means they are real. So where do we go from here? Mini brain balls. Back in April, and even a year before that, they've been tinkering with them down long, dark hallways. Well, listen to this. 15 hours ago, in D.C. this weekend, brain science meets behavioral science to shed light on mental disorders. Now, we've talked about this 21st Century Cures Act and all this funding going into treating mental disease. Tommy Insel leaving the head of the National Institute of Mental Health and going over to Google's Verily division, which I just saw recently he left. And I'm not quite sure where he is now. But he was talking about being able to come up with quantitative methods of diagnosis for mental disease, such as diagnosing your disease by the sound of your voice. And they'd been working on smartphone applications to be able to do that. Well, now we have this big gathering of 30,000 brain scientists in Washington, D.C., attending the Society for Neuroscience meeting. One of the hot topics this year is mental disorders such as depression and schizophrenia, as well as autism. And NPR was there, and their correspondent, John Hamilton, just came from a meeting to talk about some of what he is seeing and hearing. So they're going to ask him a few questions. So how does this work contribute to the understanding of mental disorders in people? And the report, he says, 20 years ago, I'd say it didn't contribute much, but things are really changing. I was really surprised. I was going through the abstract to this year's meeting, and there were nearly 1,000 papers that mentioned depression. There were 500 that mentioned schizophrenia or autism. And just this morning, there was a study on how looking at the brain tissue of people with obsessive-compulsive disorder and how it looks different. So the fields of brain science and mental health are converging. And I think the reason is that brain scientists are finally beginning to figure out how the biology works and the biology that underlies mental health problems. So as they're creating mentally unstable AI and then genetically defective mini brain balls, they're discovering the biology. So I was talking to a scientist at this meeting. His name is Robbie Green. He's a psychiatrist, but he is also a lab scientist at Southwestern University in Texas. And he was telling me that neuroscience is now at a point where it can help psychiatrists and psychologists understand all those things that are happening in the brain that we are not conscious of. Robbie Green says, now that we're starting to actually understand this unconscious activity in terms of how the neurons are working, firing, talking to each other in ways that produce behavior and feelings, things that we can actually see. So has something changed to let scientists understand so much more about what's going on in the brain? And he says, well, a lot of things have changed, but one of the big things is the ability to reproduce aspects of human mental health disorders in animals. And if you do this using genetic engineering, there's a technique called optogenetics. Now, we're going to talk a little bit about optogenetics. And that it lets scientists use flashes of light to switch on and off specific brain circuits in animals. Not a person, he says, wink. But this is how Robbie described what it can do. You shine this light on a specific area of the brain and activate those neurons. And all of a sudden, this mouse that we were doing our research on changes his behavior. And you say, oh, here's a circuit that is controlling whether an animal, let's say, avoids something or goes toward it. I should say science has been using this technique to induce things like depression and anxiety. So you can turn that switch on, and in the mouse, he gets depressed. You can turn it off, and the mouse feels better. And it gives you a way to identify the circuits that are involved in depression. So they're going to start using optogenetics to treat mental disorders. And this also gives you a way to see how the rest of the brain is reacting to the depression. But Martin, the reporter, says that is still in animals. How do we know these things work the same way in humans? And well, he says, there are now ways to actually watch the human brain as it is working, and probably one that people best know is called fMRI. It has its limitations, and we can look at individual brain cells, but it can't tell you what certain areas of the brain become active and when two areas of the brain are communicating. And that means scientists have a way to see whether something they noticed in a mouse brain seems to be the same in a human brain. So one question people have with all of this is this all leading to some kind of new treatment for mental disorder? Well, he says, yes, of course it is. And it is beginning to. All of this stuff about brain circuits we've been talking about, it turns out to be really important for treatments like brain stimulation. So people know about brain stimulation because it's a way to reduce tremors in people who have Parkinson's disease. And it turns out that there's more and more evidence that you can treat other diseases by stimulating different areas of the brain. For example, if you stimulate one area of the brain, it can relieve some of the symptoms of depression. If you stimulate a different area, it seems to relieve some of the symptoms of obsessive-compulsive disorder. And doctors would not have known which of these areas to stimulate if it had not been for the basic research that comes out of places like the Society for Neuroscience meeting. Well, there you go, guys. Optogenetics and just flashing little bits of light to flip switches in the human brain. Let's talk about some of the research that is going on in the lab with little mousies and some of the things that they talked about at this year's meeting in D.C. I came across a story on Friday, maybe Thursday. Scientists implanted rodents with lab-grown human mini brains, and that is not even the scary part. So little mini brain balls have now been implanted in rats. Stem cells research has been a hotly debated issue for years already, but groundbreaking work utilizing stem cell-grown human brain tissue is about to challenge even the most lax definitions of medical ethics. Two groups of researchers are poised to present their work regarding the implantation of human mini brains into rodents and the potentially horrifying results that followed. The two papers, summaries of which have been reportedly submitted to their to be presented this year at the Society for Neuroscience in D.C., reveal that not only did the human mini brain tissue survive for extended periods of time, but that it actually began to integrate with the rats in unexpected and incredible ways. The tiny brain blobs, which scientists call organoids, were able to be successfully implanted from their lab-grown conditions into living mice. And once implanted, the brain tissue melded with the rodent's circulatory system, allowing it to remain alive for extended periods of time. And then here's the scary part that should really send a chill up your spine. The human brain tissue actually managed to connect itself to the host nervous system, sending tiny wires that carried nerve signals to multiple regions of the mouse's own brain. The researchers stopped short of saying whether or not this is a clear connection between human brain tissue and the rodent brain, and it caused less test subjects to behave differently, or if it appeared as though the mice had any shifts in intelligence or reasoning, and that is likely to be expanded upon once the research is formally presented. It brings up some pretty interesting questions about what allows us ethically to do research on mice in the first place, namely that they are not human. And if we give them human cerebral organoids, what does that do to their intelligence, their level of consciousness, and even their species identity? And some astute listener on the Facebook feed for this show actually left a comment about if we're giving citizenships to humanoid robots such as Sophia, what does this mean for humanized rats? Which it is true. How frightening is this, guys? I mean, this really is mad science gone off the rails. So these are questions that researchers will have to answer as they continue to consider the implications of using artificial human brains in experiments and trials such as these. But at the moment, it seems as though ethics is taking a backseat to scientific glory. So that's why we had 30,000 neuroscience and brain science researchers show up at this DC meeting. All right, so now that story was out of a publication called BGR.com. This one is out of stat.com or statnews.com. Tiny human brain organoids implanted into rodents trigger ethical concerns, because this brings up a few details that weren't in that first story. Minuscule blobs of human brain tissue have come a long way in the 4 years since scientists in Vienna discovered how to create them from stem cells, and the most advanced of these human brain organoids, no bigger than a lentil, and until now existing only in test tubes, pulse with the kind of electrical activity that animates actual brains. They give birth to new neurons, much like full-blown brains, and they develop the 6 layers of the human cortex, which is the region responsible for thought, speech, judgment, and other advanced cognitive functions. Oh no, the mice might be able to learn to talk. These micro quasi-brains are revolutionizing research on the human brain development and diseases from Alzheimer's to Zika, and the headlong rush to grow the more realistic, highly developed brain organoids has thrown research into uncharted ethical waters. Like virtually all experts in the field, neuroscientist Hung-Jung Song of the University of Pennsylvania doesn't, quote, believe an organoid in a dish can think, he said, but it is an issue that we need to discuss. Those discussions will become more urgent after this weekend. At a neuroscience meeting, 2 teams of researchers will be reporting on implanting human brain organoids into the brains of lab rats and mice, raising the prospect that the organized functional human tissue could develop further within the rodent. Separately, another lab has confirmed a stat that has connected human brain organoids to blood vessels, the first step toward giving them a blood supply. That is necessary if the organoids are to grow bigger, probably the only way that they can mimic fully grown brains and show how disorders such as autism, epilepsy, and schizophrenia unfold. But the vascularization of the cerebral organoids also raises such troubling ethical concerns that previously the lab paused its efforts to even try. We are entering totally new ground here, says Christoph Cook, president of the Allen Institute for Brain Science in Seattle. Ding, he keeps coming up in all these stories. And he's the one that is holding the brand new 3D map of brain neurons from some of the work of Georgie Church. The science is advancing so rapidly that the ethics cannot keep up. Neither can regulation. Although the National Institute of Health has a moratorium on funding research that puts human stem cells into early embryos of vertebrates, there is no such ban on implementing human organoids, and virtually no one outside of the lab conducting organoid research has enough of an understanding about what is going on, say, to call for a commission to study what should and should not be allowed. Mad science gone off the rails with no controls. In a previously unreported experimenting of implanting human brain organoids into lab rodents, that means someone might have caught them, most of the transplants survived, in at least one case for 2 months, according to the summaries of 2 papers being presented at the annual meeting of the Society for Neuroscience in Washington, D.C. And more notably, the human organoids implanted into mice connected to the rodent circulatory system, making it the very first reported vascularization. A mature neuron from the human brain organoid sent axons, which are the wires that carry electrical signals from neuron to neuron, into multiple regions of the host mouse's brain, according to the team, and from the Salk Institute, which is one of the world's most eminent neurobiology labs. The human brain organoid cannot reach anything close to the size of a child's brain in the tiny confines of a mouse, which is 1/1000th the volume of a person's brain, almost certainly limiting how complex it can become. But these little suckers are not going to say hi, says a legal scholar and bioethicist Hank Greeley from Stanford University. But the rapid advances, he says, are raising the question of whether you are creating something humanish that you have to take seriously in terms of according it dignity and respect, and figuring out what that even means. Well, very good question there, Johnny. And if those issues emerged 17 years ago when a Stanford colleague of Greeley's proposed implanting human stem cells into mouse brains to see what would happen when the former turned into neurons, the experiment has not yet happened. But the ethical consensus was that it might be okay if a fraction of human neurons in the mouse's brain was so small that the implant had no chance of giving a mouse human consciousness or enhanced intelligence. A summary of his experiment that Gage sent to the neuroscience meeting did not specify the size of the human brain organoids that he and his colleagues implanted into the mice, but he told Stat that he could not talk about the work because he had submitted it to a journal. Nor did the summary indicate whether the Salk team tested for changes in the mice's abilities or behaviors. But if the human organoid caused such changes, it would raise profound ethical concerns, says bioethicist Josephine Johnston of the Hastings Center. It brings up some pretty interesting questions about what allows us ethically to do research on mice in the first place, namely that they're not human, but if we give them human cerebral organoids, what does that do to their intelligence, their level of consciousness, and their species identity? And if another study scheduled to be presented at the neuroscience meeting, which is 21 organoid papers are now on tap. So it's 21 labs doing crazy experiments that we will probably never hear about. Researchers led by Isaac Chen, a neurosurgeon at the University of Pennsylvania, implanted human cerebral organoids into the brains of 11 adult rats, specifically a secondary visual cortex. That structure processes visual signals to, and for instance, figures out the color and the orientation of something that the eye has seen. Now, why would they do that? Well, because the color can flip switches on and off to make it depressed or have schizophrenia. The 2-millimeter organoids survived for at least 2 months, Chen said in an interview, and showed extensive growth of human axons into the rat's brain. Some of the axons grew as much as 1.5 millimeters, connecting to the corpus callosum, a bundle of neurons connecting the left and the right central hemispheres. When the scientists shined a light ray on the rat's eye or stimulated brain regions involved in vision, neurons in the implanted organoid fired. That suggested that the human brain tissue had become functionally integrated with the rats. Chen's hope is that organoids may one day be used to treat brain injury, stroke, even schizophrenia and autism, and scientists have long hoped to do that for Parkinson's disease using injections of neurons, essentially called cell slurry, which is frightening in and of itself. And think about this one. They want to start injecting cell slurry into people's brains to make things grow that they can control. My interest in repairing the brain, says Chen, and he suspects that implanting tissue that is already organized, structured, and displaying electrical activity, a brain organoid, might work better. The brain is very, very ordered, and so an implant with an existing structure and intrinsic circuitry might integrate with the damaged brain more effectively and fix it. And he and his colleagues discussed the ethics of implanting human brain organoids into rats and whether the animals might become too human. Some of what people warn about is all science fiction, he said. Right now, these organoids are so crude that we will probably decrease the rat's brain function. Ethicists argue that not a problem now doesn't mean that it will never be a problem. One concern raised by the human brain organoid implant is that functional integration of the organoids into the central nervous system of animals can in principle alter the animal's behavior or needs. The task then is to carefully monitor if such alterations occur, and if human implants give an animal increased sentience or mental capabilities, he added, it might suffer more. Would it feel like a human trapped in a rodent's body? Because both the Salk and the Penn experiments use adult rodents, their brains were no longer developing, unlike if the case of the implants were done on a fetal rodent brain. It is hard to imagine how a human-like cognitive capability like consciousness could emerge under such circumstances, but he said that his experiment carries much less risk of creating animals with greater brain power than normal because the human organoid goes into specific regions of an already developed brain. The belief that consciousness is off the table is in fact a subject of debate, and an organoid would need to be much more advanced than today's to experience consciousness, says Allen Institute Cook, including having a dense neural connection, distinct layers, and other neural architectures. But if those and other advances occur, he says, then the question is very germane. Does this piece of the cortex feel something? And whether a brain organoid can achieve consciousness without sensory organs or other means of the perceived world, or would it experience something different than what people and other animals do? It raises questions. What is it conscious of? Well, now let's take a look at a little bit of optogenetics. This is out of Wired.com from September 8th, 2015. The unexpected science of manipulating neurons with light, and there was something that really caught my attention in this story, and I wanted to point this out to some of you, especially those of us who maybe live along the Great Lakes. So, this author writing the story, Sarah Zhang, the first time I learned that scientists could control neurons with light, I definitely did not keep my cool. My response was something like, holy shit, I had no idea you could do that, unless you listened to Before the First Cup. Perhaps not the best thing to say when you're trying to convince a neuroscience professor to hire you as an undergraduate researcher. Hence, I did not get hired. That was in 2008, 9 long years ago, when the groundbreaking new technique of optogenetics was just bursting into neuroscience. Optogenetics is a matter of putting light-sensitive proteins called opsins into specific neurons using genetic tools, and the resulting possibilities still have a shade of science fiction. A flash of blue light to a certain bundle of neurons can turn an ordinary mouse into a seething ball of aggression. Or it can implant false memories, or it could let deaf mice hear again. Just a flash of blue light. Hundreds, if not thousands, of labs now use this technique. And remember, guys, we're all staring at monitors and our connected devices 24/7, where the prominent light is blue. 10 years ago this month, so this was written in 2015, so this was in 2005, Nature Neuroscience published the first paper showing that a light-sensitive protein from algae worked well at turning on rat neurons. So they are using light-sensitive proteins from algae. Now remember, we're having all these algae blooms all over, caused from climate change, quote unquote. And I know the Great Lakes, the one end near Cleveland, they had some issues going on last year. We always have had like an algae taste in the summertime, but then you get lots of chlorine in the water. But I just thought it was interesting that they are experimenting, and I would almost guess with maybe some mini brain balls, if not in mice, in the lab with optogenetics and algae. Since then, scientists have used optogenetics in mice, rats, fruit flies, nematodes, and even monkeys, and it is hard to understate the technology's impact in neuroscience. Optogenetics has more than anything else let people play the piano in the brain, just opposed to maybe slamming their whole forearm down on the keys, says Joshua Sains, a neuroscientist at Harvard. Before optogenetics, neuroscientists largely relied on implanting electrodes to control neurons, and that went one of two ways: either activate one neuron at a time or activate millions of neurons at a time. There was no way to stimulate the same group of neurons consistently, experiment to experiment, animal to animal. Plus, the electrophysiology is extremely finicky. Grad students spend years perfecting their technique. Francis Crick speculated about the way out idea of using light to activate neurons decades ago, and the need was clear, but the scientific community was initially skeptical. And remember, your brain works on different electrical impulses, and we've talked about this before. Waves are waves, whether they be light or sounds or energy. Energy can be a wave or a particle. Matter is never created or ever destroyed, guys. I'm just saying, this, I think, is a big deal, to be able to flip neuronal switches with different wavelengths of light. There was a bit of an attitude at the time that the technology was hype, but two of the most prestigious journals, Nature and Science, rejected the initial paper, but other scientists had put other opsins into neurons, and that didn't work very well. This particular protein is called channel rhodopsin 2, isolated from green algae, and it is an ion channel that opens in response to blue light, creating a current much like a neuron firing. So they're making little genetic algae machines. It worked the very first time, says the scientist Bowden, which is so unusual that he thought his promising data might just be an artifact. But when scientists discovered how easy it is to use opsin, it swept through neuroscience laboratories. You take it, you stick it into a cell, and you are off to the races, says Sains, who uses optogenetics in his lab to study circuits in the retina. In the past decade, scientists have optimized that first opsin and found new ones that can inhibit neurons or respond to different colors of light. Optogenetics doesn't solve every problem in neuroscience, and Boyd is one of the first to admit if the brain is a computer, then he likens optogenetics to a very good keyboard. What neuroscientists are still lacking is the ability to observe the circuits of the brain in action, exactly what happens when you press each key. Scientists are now working on tools to image hundreds or thousands of neurons at the time of a millisecond scale of a brain, and that breakthrough does come. It may come from somewhere unexpected. This protein came from a green algae first isolated in the soil in Amherst, Massachusetts, and microbiologists have found some of the first opsin while investigating a strange orange color in salty waters. That orange turned out to be an opsin that the microbes use to capture and store energy from the sun. And optogenetics is hardly the only game-changing discovery to come out of what might seem useless research into microbes. CRISPR-Cas9, a much-hyped genome editing tool, came from studying the immune system of obscure bacteria. There are so many unmined treasures out there, says Boyden. And yes, just like we are going to push a button and create life on the fly, per the DARPA lady. So I ran across this one. Talk about more crazy things that they are doing. This is out of the-scientist.com. I did not know about this technology. Base editing, now able to convert adenine, thymine, that is the A and T in ATCG, to guanine, cytosine of the CG. And with the arrival of a new class of single nucleotide editors, researchers can target the most common type of pathogenic SNP in humans. So this isn't just CRISPR-Cas9 editing DNA. This is single nucleotide editors, and that is converting a CA or CT to a CG. That would be flipping the code on a single level for a base pair in DNA. Base editing is relatively recent gene editing technique that swaps one DNA base pair for another, offering researchers the potential to correct harmful single nucleotide mutations in the human genome. But current classes of base editors only allow the conversion of a cytosine guanine, and that would be a CG base pair, to thymine adenine base pair, TA, rendering a substantial portion of such mutations untargetable with this approach. But now researchers at Harvard University have designed a new class of adenine base editors that can efficiently turn the AT into a CG, opening up the majority of pathogenic point mutations for editing. That means they're going to be able to splice and dice us like never before. The team reported their findings today, October 25th, 2017, in Nature. It is a very elegant study, says Andrew Bassett, head of the research and cellular operations at the Wellcome Trust Sanger Institute, who was not involved in the work. Being able to extend base editing to other types is really quite important, he says. Traditional CRISPR-Cas9 genome editing makes a double-strand break in the DNA in order to introduce insertions or deletions at targeted sites. But making this break can lead, we've read about this before, to substantial numbers of errors at the target site, such as random insertions or deletions of nucleotides. Base editing, by contrast, directly converts one DNA base pair to another without cutting across the nucleic acid. The technique, first developed by chemical biologist David Liu and colleagues at Harvard, is associated with fewer unwanted deletions or insertions and a higher editing efficiency. Current editors consist of a base converting enzyme tethered to a specifically engineered non-cutting version of the endonuclease Cas9. After a base conversion on one DNA strand, this Cas9 protein makes a mark called a nick on the opposite unedited strands, prompting the cell's DNA repair mechanism to replace the original, now mismatched nucleotide and thus complete the base pair swap. Guys, the stuff that they are working on, and we laugh about being genetically modified on the fly, but it is not funny anymore. I see that the technology exists out there, and they can get this stuff into us through food, through air, through water, through flashes of light, through harmless viruses such as the adenovirus. We read about them being able to insert CRISPR-Cas9 into a virus and then infect things with it. So you can go in and start splicing and dicing. So, all right, I'm not going to read too much more about this. This gets into how to build an adenine editor, and I don't really want to know how to build an adenine editor, but I had another story that caught my attention out of MIT News, news.mit.edu. Gene circuit switches, and there will be synthetic gene circuit switches. Synthetic gene circuit switches on inside of cancer cells and triggers an immune attack. And this would be in T cells. Researchers at MIT have developed a synthetic gene circuit that triggers the body's immune system to attack cancers when it detects signs of disease. The circuit will only activate a therapeutic response when it detects two specific cancer markers, and it is described in a paper published today, October 19th, 2017, in the journal Cell. Immunotherapy is widely seen as having considerable potential in the fight against a range of cancers, and the approach has been demonstrated successfully in several recent clinical trials, according to Timothy Liu, Associate Professor of Biological Engineering and Electrical Engineering and Computer Science at MIT, and a member of MIT's Cook's Institute of Integrative Cancer Research. There's been a lot of clinical data recently suggesting that if you can stimulate the immune system right away and you can get it to recognize cancer, that some of the best examples of this are what are called the checkpoint inhibitors, where essentially cancers put up stop signs that prevent T cells from killing them. There are antibodies that have been developed now that basically block those inhibitory signals and allow the immune system to act against these cancers. However, despite the successes, the use of immunotherapy remains limited by the scarcity of tumor-specific antigens, substances that can trigger an immune response to a particular type of cancer. The toxicity of some of the therapies when delivered as a systemic treatment to the whole body, for example, is another obstacle. But what is more, the treatments are not successful in all cases. Indeed, even some of the most successful tests, only 30 to 40% of patients will respond to the given therapy. As a result, there is now a push to develop combination therapies in which different but complementary treatments are used to boost the immune response. For example, if one type of immunotherapy is used to knock out an inhibitory signal produced by cancer and the tumor responds by unregulating a second signal, an additional therapy could then be used to target this one as well. Our belief is that there is a need to develop much more specific targeted immunotherapies that work locally at the tumor site rather than trying to treat the entire body systematically. Secondly, we want to produce multiple immunotherapies from a single package and therefore be able to stimulate the immune system in multiple ways. The circuit, which can be customized to respond to different types of tumors, is based on simple AND or AND gates versus AND or NOR used in electronics. So they're building actual circuits out of synthetic cells. Such AND gates will only switch on a circuit when two inputs are present. Cancer cells differ from normal cells in the profile of their gene expression, so the researchers developed synthetic promoters, which are DNA sequences designed to initiate gene expression, but only in cancer cells. The circuit is delivered to the cells in the affected area of the body using a virus. Ding, ding! And the synthetic promoters are then designed to bind a certain amount of proteins that are active in the tumor, causing the promoters to turn on. Only when these two cancer promoters are activated does the circuit itself actually switch on, says Liu, and this allows the circuit to target tumors more actively and accurately than existing therapies, as it requires 2 cancer-specific signals to be present before it responds. Once activated, the circuit expresses proteins designed to direct the immune system to kill the target of the tumor cells, including surface T-cell engagers, which direct T-cells to kill the cancer cells. The circuit also expresses a checkpoint inhibitor designed to lift the brakes on T-cell activity. So when the researchers tested the circuit in vitro, they found that it was able to detect ovarian cancer cells from amongst other non-cancerous ovarian cells and other types of cells. They then tested the circuit in mice implanted with ovarian cancer cells and demonstrated that it could trigger T cells to seek out and kill cancer cells without harming other cells around them. Finally, the researchers showed that the circuit could be readily converted to target other cancer cells. We identified other promoters that were selective for breast cancer, and I just saw a study that they think they may have now discovered an early test for detecting ovarian cancer, which is something that typically doesn't have any signs or symptoms or tests for it, and that was using some sort of genetic test. And I found it interesting that they were targeting the BRCA1 and BRCA2 genes, which are the cancer genes that are involved with breast cancer or some genetic breast cancers as well. So ultimately, they hope they will also be able to use this system to target other diseases such as rheumatoid arthritis, inflammatory bowel disease, and other autoimmune diseases. This advance will open up a new front against cancer, says Martin Fressenegger, a professor of biotechnology and bioengineering in Zurich, Switzerland, who's not involved in the research. First, author Lior Nisum, whom pioneered the very first genetic circuit targeting tumor cells, has now teamed up with Timothy Liu to design an RNA-based immunomodulatory gene circuit that takes cancer immunotherapy to a new level. The design of this highly complex tumor-killing circuit was made possible by meticulous optimizing and integration of several components that target and program tumor cells to become specific prey for the immune system, and it is a very smart technology to be in use someday through the medical establishment. This work is supported by the National Institute of Health, the Department of Defense, the Defense Advanced Research Project Agency, more affectionately known as DARPA, and the Cook Institute as well. All right, guys, have an awesome Monday.